Hello everyone, welcome to Gebze Technical University Department of Bioengineering webinar series. This is our 17th webinar. Our guest for today's webinar is Nimet Yıldırım TİRGİL from Ankara Yıldırım Beyazıt University. And the webinar title is Nanotube-Based Biosensor System for Pathogen Detection. Let me introduce our guest shortly. Assistant Professor Nimet Yıldırım TİRGİL obtained her bachelor's degree in 2017 and master's degree in 2009 from Ege University, Department of Biochemistry. She completed her PhD in the Department of Bioengineering of Northwestern University in 2016. Afterwards, she completed her postdoctoral studies in Jenner Lab. Since 2018, she is working as an assistant professor at Ankara Yıldırım Beyazıt University, Department of Biomedical Engineering. Hocam, you can start.
Okay, thank you so much for this kind introduction, and thank you so much again for inviting me in this webinar. So today, let's try to share first. Today I will try to talk about a part of my PhD work. It was actually a little old. It was from 2015, I guess. Okay. Yeah. So I will talk about a part of my PhD work. It is nanotube-based biosensor system for pathogen detection. So in this presentation, I will just try to introduce briefly biosensor systems and our nanobiosensor device and the new fabrication process. And then I will talk about pathogen detection, one virus, one bacteria detection as an example that we develop in this system. So biosensors, actually in the classes, I'm just teaching a biosensor classes. And in these classes, generally, I just try to explain biosensor as a mimic detection system for living organism. Like in our tongue, we are just sensing the taste. And in our tongue, we have receptors, the analyte bind the receptor, and then we taste different kind of food. So biosensor just trying to mimic this kind of system. So we have biorecognition molecules, antibodies, enzyme, or nucleic acid-based biorecognition molecule, or even cell. So we use these biomolecules as a recognition molecule because they just bind their target molecule 100% selectively and sensitively. So also in the biosensor system, we have a transducer mechanism. This mechanism just try to convert this sensing, this attachment between the biological molecule and target to a readable signal. This transducer system can be electrical, electrochemical, optical, colorimetric, or mass sensitive, or also there is a bunch of other transducer mechanism. So the biosensor is really combining of biomolecule transducer system. So they are selective and sensitive. Also, they have fast response because this binding, like antibody antigen binding and sensing, it is also fast. And they are cost effective comparing with the other large analytical devices. Of course, they are cost effective and they are capable to make a real-time detection because most of them is small and we can just carry them anywhere we want. They are portable, so they are just good for real-time and on-site detection. So the biosensor system, we have different kind of detection system and different kind of biorecognition molecule. Each of them has their advantage and disadvantage. So in this presentation, I will talk about electrical and conductance-based detection system. We just develop a nano device capable to make this kind of detection. And we test nucleic acid-based biorecognition element and antibody-based biorecognition element. So if we just check just briefly their advantage and a little disadvantage and challenges, this electrical conductance-based transducer, they have low cost, they are just cheap, and they are easy to use and they are fast, and also they are compatible with biomems and even with bionems system, this microelectromechanical system. And the antibodies and nucleic acid-based recognition element, the antibodies has some challenges because their structure is a peptide. They are just have antibodies in it. They have amino acid in it. And this amino acid-based molecule is generally too sensitive. They are sensitive for harsh conditions like high and low pH. They are sensitive for temperature. So because of this, if we need to use antibodies as biorecognition element, we should keep our system in 4°, or we can just use them in pH 7 to 75 kind of range, because the antibodies themselves, they are sensitive, but they are too sensitive for their target molecule. They have like 100% binding capability for their target molecule. But the nucleic acid-based biorecognition element is more stable than antibodies, because the nucleic acid, they're just quite stable. Even for 100 and 1,000 years, they cannot denature. So because of this, the nucleic acid-based biorecognition element has this kind of advantage over antibodies. So we would like to just test two of them to see their differences, their usability in this nano device. So the objective of usage of a biosensor system, as I just already told them, I guess. So they are cost-effective, real-time, on-site detection. Also in biosensor system, we are trying to leverage the current advance in biotechnology and nanotechnology field. So we are just combining biotechnology and nanotechnology and try to make a new detection system. So when we just check our nano device, actually this nano device developed in our university, Northeastern University, and we have a nano center, Egan Research Center, and the Ahmet Busnayne, he was my kind of co-advisor. He was the head of this center, and they developed this system by themselves. Actually, I just observed them, I can say, I just observed them. I never did this system by myself. I just helped them, try to observe them. So I am not very experienced in this practical nano device fabrication, but they did this fabrication in our system, and it's a new method. They are just develop a new method, and with this new method, they just develop this nano device. So actually in the device itself, we have two gold electrode, and between these two gold electrode, we have a carbon nanotube bridge. So with using a 2-point deduction, we can calculate the conductance and resistance between these two gold electrode. It's actually a basic system. You can see this system in most of the electrochemical biosensor system, but their carbon nanotube bridge development is new, and it's a novel system. Because they just use this kind of template. They are gold. They deposit this gold on the top of the silicon layer, and then they put the carbon nanotube onto this gold template. So they just deposit this gold in the desired location, and then they grow the carbon nanotube onto this gold, and after this deposition, they just transfer this carbon nanotube onto a flexible material, which is our nano device. So with this newly developed system, they could make a more uniform and high density carbon nanotubes. So this is our carbon nanotubes between these two gold electrodes. So this system is new, so they just have a published paper. They call this offset printing. So this is our system, and this is just a real image of just two gold electrodes, between them this tiny carbon nanotube bridge. Actually, our system is just on the top of this tiny places. In the small part of this flexible biosensor system, we have 48 of them. 48 of the nano device just locating in the small places. So we immobilize our biorecognition element, antibody, or in the other case, probe DNA, on the top of the carbon nanotube. We just bind them on the carbon nanotube, and we are just making a conductance-based deduction. And the system itself has a low power consumption, so it is portable. Not at this point, we didn't develop a portable product yet, but it is available to make it portable. So we have a highly aligned carbon nanotube bridge between these two gold electrodes. So it is enabled for low detection limit, because we have a dense carbon nanotube bridge, and carbon nanotube bridge has this kind of three-dimensional shape. So think about all the around of the carbon nanotube bridge, we have attach of our biorecognition molecule. And because of this high dense sensing surface, we can make a low detection limit of detection. This is the basic for our sensing principle. Electrode between them, we have a carbon nanotube bridge, and we attach our probe DNA or antibody on the top of the carbon nanotube bridge, and we got an IV measurement. So this is just voltage and current, and with using this values, we can calculate the conductance and resistance, also device itself, just conductance and resistance. And then we just add our sample, the antigen or aptamer bind to the probe DNA, and then we dry the surface, and then we did another measurement. So when something bind onto the carbon nanotube bridge surface, the conductance decreasing and resistance increasing, because the carbon nanotube bridge has the superconductance ability, but if something bind on it, its conductance is decreasing, so resistance increasing. So these differences between before and after injection, we can get the differences value, and it is just giving us the bounded analyte on the top of the system. Actually, in the immobilization part, we also have a novel side in our detection. Generally, in the biosensor system, they can immobilize the recognition molecule on the top of the carbon nanotube bridge with using a covalent binding. They just bind them directly to the carbon nanotube bridge surface, but also the carbon nanotube bridge, the shape and structure has its unique properties. It is also giving the superconductance feature to the carbon nanotube bridge. So because of this, we don't want to make a covalent binding. We don't want to make just a direct binding. So we use this kind of linker. It is pyrene butanoic acid. In the pyrene molecule, they have this benzene ring, and they have pi orbitals on it. And also carbon nanotube bridge, they also have the same shape of pi orbitals. And between them, they have a pi-pi stacking. Also in the literature, they also use this in some part. We also get their procedure. So between the carbon nanotube bridge and this pyrene molecule, they have a pi-pi stacking. It is not a covalent binding, but it is quite stable and strong attachment. So with using this attachment, we are not affecting the carbon nanotube structure, but also we have a strong binding between the biorecognition molecule and carbon nanotube bridge. Okay, this is our immobilization part. So this is our aspect of improvement. So we are trying to make a nano biosensor which will be more stable, reproducible, and uniform. And then we are just trying to make more sensitive device with using superconductive carbon nanotube bridge. So we are using a kind of novel immobilization to make our system more sensitive. And so this is label free. I will just show you the sensing system. You may understand just better. It's a label free detection. Our target, like pathogen, just bind onto the aptamer or antibody, we can make the detection. So it is label free. We don't need any labeling, like fluorescent and colorimetric kind of labeling. We don't need anything like this. Also, it is suitable for miniaturization. We didn't do this yet, but they are available for integration with biomems or bionems system. First of all, we try to detect adenovirus. It is a pathogenic virus. It is widely infect about millions of people worldwide. It is not as lethal as coronavirus, of course, but it can affect a million of people every year, and it can also can make infection, the respiratory infection in kids, and it can be deadly, especially in the kids. It is a waterborne virus. It means it can contaminate the water, and it can just infect people by water sources. So detecting this virus in water sources, so because of this, it is quite important. So there is such current detection methods for adenovirus. The most common one is cell culturing, like they just grow a cell medium and they infect them with the contaminated water. And then after 1 or 2 weeks, they can just observe the adenovirus growing. And then they just count them. It is quite similar as the plate counting, but it takes 1 or 2 weeks, and they need a laboratory equipment, of course. And the other technique is PCR, as we know widely, as we can just hear them widely nowadays. PCR is quite sensitive and also rapid. I mean, kind of rapid when we compare with the cell culture. We can make the PCR in a couple hours. They are quite sensitive, but they need DNA extraction. So we need to extract the DNA inside from the virus, and then we need to purificate the virus DNA, and then we can make the detection. So, and then they also have they also need the laboratory equipment too. So in the adenovirus, in the outer shell, they have the hexon protein. It is just even quite look like the coronavirus, the hexon protein. And we are just aiming to use an antibody directly bind to this hexon protein and grab the adenovirus from the sample, and then we try to make a direct detection. So compare with them, our system can be more comparable, sensitive, and definitely fast and direct detection. Also, we just know that no direct detection of live adenovirus in the literature so far. So our system will be a model virus detection in real samples. So we applied this system for adenovirus, but it is just available to test the system for any other virus. This is our sensing mechanism, our flexible biosensor. They are our gold electrode and carbon nanotube bridge. So we immobilize antibodies, the hexon antibodies, directly on the top of the carbon nanotube bridge. And then we use a non-covalent binding again. After this immobilization step, this system will be stable in 4 degree, of course, in 4 degree, because we have antibodies on the top, 4 degree for months. After we first make this immobilization and a preparation of the nano device, we can just store them, and whatever we need, we can just get and just reuse it, okay? After the immobilization part, we just inject the water sample on the top of the device, and then the adenovirus bind to the antibodies. Actually, this is not on scale, you know, the antibodies is much more smaller. And think about, we have the antibodies all around the carbon nanotube, three-dimensional shape, all around is just full of antibodies. And in the literature, they said there is about up to 200 hexon protein on around the adenoviruses. So think about at least 100 of them, 100 of the antibodies grab one virus. So it is a quite stable attachment, quite strong capturing. So after they capture them, we wash the surface just with PBS, try to wash away the impurities coming from the water sample, and then we did the IV measurement. Just before injection, after injection, and differences between them give us the signal coming from just the adenoviruses. So after we test the system, we try to regenerate the surface. So we try to cleavage the virus from the antibodies and reuse the clean biosensor surface again and again. So we try to optimize this. It is not very optional for antibodies because they are too sensitive. The regeneration solution can affect them, but we could make this. We just realized that we can use the same sensing surface up to 10 times with this regeneration. So it was also good. This is our summary of the result. So we are capable to make adenovirus detection in the 10 PFU per ml to the 10 to 6 PFU per ml linear range. And this is our percentage of ST, the standard deviation, percentage of standard deviation. Also, it is not bad for a direct virus detection up to 20% false result. It is also acceptable in the biosensor field. This is our specificity and selectivity result. So in our system, we test such the other pathogenic bacteria and viruses, and we use this range 2000 PFU per ml. Actually, I should say that this is too high, like 2000 PFU in ml, it is too high, but we would like to test this much high range because the adenovirus amount can be very small in a water sample, but in the sample, there must be such the other kind of pathogenic or non-pathogenic bacteria and viruses. So because of this, we try to test this much high range, and our system has quite sensitive and selective result for adenovirus. Such the other virus give less than 20% result with our system, which is also acceptable and good for biosensor. And this is our electrode stability, I mean the biosensor surface, nano-device surface stability. After 5 days, they are even usable. Actually, this is the reusage. I mean, in the first day, we use the same system. The third day, fifth day, we use the same system surface again and again. And this is our regeneration result. So we try to test different regeneration solution. Actually, this solution has higher and lower pH. This binding only happen in a distinct pH, actually in 7 to 7.5. If we change the pH in the environment, they get cleavage from each other. So inject a lower and higher pH solution in the surface, try to cleavage the adenoviruses from the antibodies. But because the antibodies is quite sensitive, like this harsh SDS and hydrochloric acid, just denature the surface, but we can get a good result with 0.1 millimolar sodium hydroxide for 2 minutes incubation. After 10th usage, even we have 80% of the signal. So at the end, we just test the real water sample. The plant 1 and plant 2 means it's just real wastewater coming from wastewater plant. And this is the tap water. Because this wastewater don't have any adenovirus contamination, we spike the adenovirus in it. We add this much adenovirus in it, and we are just trying to make back calculation, back detection. And this is our recovery. So you can just see that we have about 20%, 25% false result, which is also quite acceptable. Maybe you can just know that in the literature, it is even say that for PCR result, they may have 30% false result. So in the other part, we just try to make a pathogenic bacteria detection, just the whole bacteria detection. And in this case, we use aptamer, which is a synthetic biorecognition element. We just use them. So here we select E. coli, this infectious strain of E. coli. We just select this because it is an indicator molecule. So it is an indicator molecule for fecal contamination. So if we are assuming there is a contamination in a water source, it should be too costly and time consuming to test every individual pathogen. So because of this, we have some indicator organism. So the total coliform bacteria or fecal coliform bacteria indicators, which is E. coli, this infectious strain of E. coli. So it means if we detect E. coli in this water source, it means there is such the other pathogenic bacteria in it, and there is fecal contamination in this water source. Also, there is such the other traditional detection methods for E. coli. The most common one is colony counting. They just get the sample and put them in this agar plate, and they just grow the E. coli in it, and then they count the colonies, but it is long. It takes at least 24 hours, and it needs the lab environment. And PCR, again, extraction of the DNA and making the DNA detection, they're fast and sensitive, but as I said before, they have some challenges too. Also, there is such ELISA systems, enzyme-linked immunoassay systems. They use antibodies like us. They are not too stable for a long time, and they need laboratory equipment again for ELISA system, and they should use this system, they should keep the system in 4-degree indicator organisms. For the indicator organism and for the pathogenic bacteria, we can just say that even one single bacteria can be contamination, because they are living organisms. They are just growing time by time. So because of this, the EPA, Environmental Policy Agency, said the standard for drinking water should be lower than the 1 CFU per ml. It means there will be no pathogenic bacteria. For streams and lakes, it should be 20 CFU per ml. So it means our system should be super sensitive to detect the pathogenic bacteria. But in this case, we have some challenges to detect this much sensitive pathogenic bacteria because especially in the water sources, there is such the other pollutants, but our pathogenic bacteria level should be very small. So it is hard to decrease the matrix effect. So it is one challenge. And sensitivities and other challenges. Also, there is high level of false positive and false negative result. It is another challenge for all the pathogenic bacteria detection. And pre-experiment is another challenge. So if you need to make any pre-experiment, it means you should get your sample from a lake and you should carry this to the laboratory. But at this time, your bacteria is just growing because they are living organism. So there is this kind of challenges. So because of this, we try to make a sensitive, person-sensitive, portable, and also easy to use system. And also we try to improve the detection level to reach as much as possible to the EPA allowable level. To make our system more stable, even stable in any condition, we try to use DNA aptamers, which is a synthetic biorecognition molecule. And also in our system, we try to make an indirect detection mode. So I will just explain them with the shame, you may understand just better. But this our indirect detection mode also capable to make our system more stable and sensitive and short, fast detection. So at this point, I would like to talk about a little aptamers. So aptamers are synthetic single-strain DNA, RNA, or nucleic acid molecules. They have highly specificity for their target molecule, like the same as the antigen-antibody binding. The aptamer, the single-strain DNA, get their 3D formation and they bind their target molecule. They are even widely used in targeted delivery of the drug. You may just see this kind of paper in the literature. The aptamers just bind the biomarker in like a cancer cell, so they recognize the cell. So they have quite sensitive binding for their sensitive and selective binding to their target molecule. But they are much more stable than the antibodies because they are DNA. They are just a short single-strain chain of DNA, and they never denature in any harsh condition. And also they are more smaller than antibodies. So with using the aptamer, we capable to make much dense sensing surface. So we can immobilize a larger amount of antibodies on a detection surface. And they are easily modified with their terminal site. We didn't make any modification in our system, but if we need their 3' or 5' easily modified with different kind of functional group. So this is the aptamer's advantage. So we are trying to use them to make our sensing system more effective. This is selects the synthetic aptamer selection process. This is a in vitro process. So at this part, I will just try to briefly try to make introduction of this. The nucleic acid pool means 10 to 14 size of different aptamer sequences. We just prepare them synthetically in a laboratory. So we mix this large nucleic acid pool with our target molecule. And some of them bound, some of them not bound. So we just remove the unbinding aptamers and we just continue with the bound one. And we just repeat this cycle again and again up to 10 and 16 times. And after we select all the bounded aptamer, we are just trying to make the characterization experiment to them, and we just try to detect the KD values, the coefficient of dissociation, the binding efficiency coefficient. We are trying to test them, and then we just select 1 or 2 or maybe 3 best aptamers. So we just said that this aptamer specifically and selectively bind our target. So we just synthetically select the aptamer molecule, a synthetic biorecognition molecule for our target, and we start use this in biosensor system. So this is our E. coli detection process. Again, we have the same biosensing surface, and then we immobilize the probe DNA. This probe DNA has the complementary sequence to the aptamer sequence. The aptamer, I will just show this here. The aptamers, these aptamers specifically bind the outer surface of the E. coli. In the E. coli, they have a lipopolysaccharide in the outer shell, and this DNA aptamer just bind them. So this DNA aptamer bind to the whole pathogenic bacteria surface. So we don't need to make any DNA extraction or anything like this. They are bind directly to the pathogen surface. So in a separate place, we mix the E. coli cells and aptamer. And then after this binding for a while, we filter this mixture to the 0.22 micron pore size filter. So the E. coli cell has the dimension of diameter of 0.4 to 0.5 micron. So they couldn't pass through these pores, these small pores. Just the unbound aptamer, there's just non-bound aptamer. Some of them bound to the E. coli. They just stuck on the top of the filter. And then we pump this sample, this unbound aptamer, to our sensing surface. So they hybridize each other because they are complementary to each other. They just hybridize each other, and then we make the IV measurement. We try to calculate measure the conductance before and after injection, and then we perfectly regenerate the surface because the DNA just immobilized on the top of the surface is super stable. They not denature with any harsh conditions. Just the high pH of SDS cleavage this hybridization. They don't denature the surface. So we regenerate the surface, use the same surface even up to 100 times. So this is our detection procedure. With this way, they are not portable. They are not capable to make portable. We have lots of, the addition, mixture kind of step, but it is capable to integrate to a microfluidic system, the mixing in a microfluidic system, pumping through a microfluidic system, binding, regeneration. So it is quite capable, has a capability to integrate with the biomems system. This is our result. We are capable to make, actually, experimentally, we can detect 2 CFU per ml of E. coli, but it is not our linear range. So we just prefer to say we are capable to make 10 to 10 to 5 CFU per ml E. coli because this is our linear range. And this is our percentage of coefficient of variation result, percentage of standard deviation, a little high to the 22%. This is our selectivity and specificity of the sensor. So we test like the other pathogenic bacteria in our system. And again, we test 10 to 4 CFU, which is quite high amount. Because, you know, in a contaminated water, there must be such the other pathogenic and non-pathogenic bacteria and viruses. So we test this much high detection level. And this is our control experiment. So in this control experiment, we have only aptamer. So this is giving us the pore, the filtration effect. No bacteria, just the aptamer, and we just filter them through the 0.4 micron size filter. So it is just giving us this amount of aptamer on the top of the filter without any bacteria. So it is just negligible because it just gives us less than 10% result. And the other bacteria, this one is just the highest one, about 25%. But as I said before, it is even still acceptable for this kind of biosensor systems. Electrode stability. We test the same sensing surface, the same nano device again and again through the 30 days and 5 different concentration every day. So still we have even 80% stability of the surface. And again, we just try to make spiked sample detection. The sample getting from the environmental wastewater plant, and we test the tap water. Again, you can just see that we have about 20% false result. So we test 10 and 10 to 4 CFU per ml level. So I guess this is what I have for our work. And also this is our patent application for this work. We just start applying the patent in 2015, and finally we could get in 2018. This is our patent application. And thank you so much for listening, and I will be happy to accept any question.
Thank you for your nice presentation.
Okay.
Now we can have we can take questions.
Yeah, definitely. No question. Okay, I could just explain everything good enough.
I think we have no questions.
Okay, nice.
Yeah, I have a question.
Uh-huh.
I am very. I like to ask that while you are developing biosensor, you mentioned that you set up some maybe antigen or peptides in gold surface layer. Did you develop this mechanism, or you buy this mechanism as a ready, as a commercial?
Okay, I've just showed. Yeah. This nano device was developed from our a kind of collaborator in Northeastern University. We have a nano device center, a kind of I'll just show this slide again. My co-advisor, Ahmet Busnaina, he was the head of this department, and they developed this system by themselves, but by using a new technology. So they developed this system. They just developed the nano device by themselves, and then I just observed them while they are fabricating this. I know the theoretical part, I'm not expert in the practical fabrication part, but they develop this, they fabricate this nano device, and they just give us, and then we apply this to this biosensor.
Okay, thank you very much.
Thank you.
Hello, I also have a question, if you have a minute.
Yeah.
Hello. I was wondering how the infrastructure is in Ankara. I mean, are you able to continue in the same line of research, or did you have to adapt a bit based on the infrastructure available to you right now in Ankara?
Actually, in our university, we don't have any nanofabrication center. But, you know, in UNAM, the Bilkent University, they have this quite available laboratories. We can even use their laboratories, but I am not continuing this research. And maybe in the future I can continue, but I definitely quit this work for now.
Okay. Okay. Well, would you be able to tell us a bit about now your new interests then?
Actually, nowadays, I am just trying to conduct a project for aptamer selection for a new target. You know, the aptamer selection, the select cycle. I am just trying to make the aptamer selection for a new target and try to develop an electrochemical detection system with using just the ready-to-use electrode. I am just trying to start this project. And also, I am just interested in portable colorimetric biosensor system. Actually, you know, more cheap, not this nanofabrication, more cheap. And also, I'm just interested in portable, ready-to-use kind of biosensor system. But I am still working on aptamers because I like aptamers. They can use it. Yeah.
Okay. Thank you very much. Thank you.
You're welcome.
Do you have any other questions? If not, I would like to thank everyone who joined this webinar. I hope you enjoyed. See you in the next webinars. Thank you for joining.
Thank you. Thank you so much.
Thank you.
Have a nice day. Bye.
Goodbye.
Hocam çok teşekkürler. Görüşmek üzere. İyi çalışmalar.